Okay, so welcome everybody to the people who already joined. Thank you for being here for the new session of the BioClock Academy lecture series. Before we start, I would like to say, yeah, this is recorded. You already heard it in the announcement. You can still leave your camera on if you would like to. It's not going to be recorded, but so yeah. Nice to see you here. My name is Laura Paap, and I'm a PhD student at the BioClock Consortium. Together with Georgiana Roos Oswald, postdoc at the same consortium, we will host the session today. For those of you who are new here, the BioClock Consortium is a Dutch research organization, and it aims to do research on different aspects of the biological clock in humans, in animals, in health and society. And we organize this lecture series for any early career researcher or just anyone really who's interested in the field of chronobiology. With the aim to bring to you basic concepts and theoretical knowledge by expert speakers in the field of chronobiology. And we do this series once a month. Today's session, we will have a 40-minute talk and 10 to 15-minute discussion. If you have any questions during the talk, if any questions come up, just put them in the chat and we will read them out loud after the talk, or after the talk, you can just unmute yourself and also ask questions personally. And now I give the floor to Georgiana, who will introduce our speaker today.
Yeah, hello, also from my side. Our today's speaker is Frank Scheer. He is a professor of medicine at Harvard Medical School and the director of the medical chronobiology program at the Brigham and Women's Hospital in Boston. Some of his main research questions are: what is influencing the endogenous circadian system and how is it disrupted, for example, by shift work or late night eating? What are the effects on cardiovascular, pulmonary, and metabolic regulations, but also how are they linked to disease states such as hypertension, asthma, obesity, or diabetes? And understanding better the biological basis of these conditions would strengthen the development of personalized interventions to prevent, treat, or manage the disease. But to not take further too much of your curiosity and let just Professor Scheer explain this, I want to give him a warm welcome. We are very looking forward for your talk. Thank you for being here, for you in the morning, for us in the afternoon, and the virtual stage is yours. Welcome, Frank. Thank you.
Great. Well, thank you so much, Laura and Georgina, for the kind introduction. Good afternoon and good morning. Goedemiddag. It's great to be here and really delighted to be invited. I see some familiar faces. Great to see you. Thanks for joining. So I'll be presenting primarily on circadian misalignment and chrononutrition today, and primarily focused on the human system. And I'm really thankful to the previous speakers for introducing a lot of the concepts already, which will make it easier, hopefully, for the audience to follow along. But if you have any questions, please don't hesitate to interrupt me or put your questions in the chat. So this is the overview of my presentation today. I'm going to very briefly talk about the circadian system because hopefully that the basics are well understood by the audience. And then I'm going to discuss the main parts, which are about the potential causal role of circadian misalignment in helping explain the well-known adverse health effects of shift work and other circadian rhythm sleep-wake disruptions. Then I'm going to talk about the important role of meal timing. So not just what we eat, but also when we eat, and focus on both glycemic control and energy balance control. And then in the last part, I'll bring these last two concepts together. So can we use meal timing as a countermeasure in the fight against the adverse effects involved with shift work? So it's the circadian system. I want to highlight this seminal work that was done by Nathaniel Kleitman, who's also known as one of the grandfathers of sleep medicine, but also played a key role in understanding and uncovering the importance of the circadian system in human physiology regulation. So here's a picture of Nathaniel Kleitman with his colleague, where they spent many days and weeks in the Mammoth Caves in Kentucky, and they measured carefully each other's core body temperature. And what you can see here on this plot is, should not surprise you, which is that core body temperature is high during the waking hours, indicated here in the light color, and the dark colors are indicating sleep. So we have about a week or so of a sleep-wake cycle. You can see that temperature drops during the sleep episode when people are living on a 24-hour sleep-wake cycle. But what was surprising was what happened when they started living on a 28-hour sleep-wake cycle. So they woke up 4 hours later every cycle. And you can see here the temperature of one of them. And what you can see is that it starts off, as you would expect, high temperature during the wake episode and low during the sleep episode. But that's something very strange happening halfway through, which is that the highest temperature is actually observed when the individual was sleeping, and lower temperature while awake. And then at the end of the protocol, it kind of reverts back to what you would typically see. So how can this be? Well, this becomes more clear if you actually draw these guidelines at every 24-hour mark. And what you can see is that in addition to effects of the sleep-wake cycle, which certainly there are, you can also see that there's something else that is maintained at approximately a 24-hour cycle. So I hope you can see my cursor here. Yes, you can. Great. So you can see that there remains to be an influence even here when people were sleeping, that when this person was sleeping, the temperature was high. And so this something is the influence of the endogenous circadian system, which ticks along at a rate of about 24 hours every cycle. So that's referred to as the period, which is the cycle length. So this was really an eye-opening experiment that was done. And since then, we have gained a lot of insight into both the molecular and neuroanatomical underpinnings of this system. Of course, in the early 70s, the important discovery of the suprachiasmatic nucleus was key. So in about 1972, by different groups. And so this was discovered by placing lesions in different areas of the brain, because people thought, well, it must reside somewhere in the brain, this clock. So in rodent studies, they made these lesions and found that if the suprachiasmatic nucleus is lesioned, these animals became arrhythmic. This is abbreviated here as SCN. But then subsequently, what was also shown is that if you remove this SCN, the suprachiasmatic nucleus, and you place it in a petri dish, this SCN itself has the capacity to oscillate with a rhythm of about 24 hours. So this is a time-lapse video. So each fluctuation shows one 24-hour cycle passing. So these two levels of evidence then show that the SCN is both necessary and sufficient for circadian rhythm generation. Now, necessary for the animal, the readout here was locomotor activity. And we can go back to that at some later stage. But then about 20 years later or so, it was realized that the same molecular machinery that underlies circadian rhythm generation in a cell, so creating a cell autonomous clock, that these transcription translation feedback loops also were present in virtually all organs and tissues throughout the body. So you have these clocks in your heart, in your pancreas, in your liver, even in your skin and fat cells. Now, that may seem like an incremental discovery, but it was actually a watershed moment, because it changed the system from a single clock system to a multi-oscillator system. And not only that, but also the discovery that these various clocks were actually responding to different time cues or zeitgebers. So as you have learned, from the previous lectures, light is the most important time cue for the SCN, and this is all mediated through light exposure through the eye in humans and most mammalian species. But what also was discovered, and I'll circle back to that later on, is that organs like the liver are very sensitive to the timing of food intake. To such an extent that if you give food at an unusual or atypical time, you can shift the liver independently of the central clock, independently of the SCN. And so it may be that your SCN is in Leiden, but your liver is in Boston, just by the mere fact of when you choose to expose yourself to light and when you expose yourself to food timing. So with that brief background, let's jump into what happens when actually there is mistiming of either the SCN or other components of the circadian system with each other or with behavior or light exposure information. So this is summarized here in this review we wrote some years ago, which is that circadian misalignment, and you may hear this word used quite often. Some people refer to circadian desynchrony, which is actually a different, technically speaking, it's a different beast, but it's often used interchangeably. We can discuss that later. But what I want to highlight here is that you can actually have many different flavors of circadian misalignment. You can have misalignment between the SCN, the central clock, and some peripheral clocks, let's say in the liver or in the lung, which we refer to as internal circadian misalignment. You can also have misalignment between the SCN and the light dark cycle or relative to certain behaviors. But you should also recognize that you can have misalignment with one behavior, but not the other behavior. So because we can, as humans certainly, but also in experimental animal models, you can dissociate the timing of food intake, for example, relative to that of locomotor activity. And I will show an example of that a little bit later. And then there are more, even more fine-grained misalignments you can have, for example, in neighboring liver cells, where they are not well synchronized and they may run on different time zones altogether. So why is this important? This is not just an academic exercise. This is something that may have real life consequences and relevance, given the fact that shift workers, when they engage in this case, this example here in A, rotating shift work, their central clock doesn't necessarily synchronize to their imposed work schedule. In fact, in most cases, it will not synchronize, and in most cases, it will actually remain remain synchronized to the solar day. So what's indicated in these circles here? So the work shifts are these blue areas, but the circles here indicate the melatonin peak levels in these individuals. I think this data was from urinary 6-sulfathoxymelatonin assessments, but if you would measure plasma, you would find the same pattern. So what you can see is that despite the work schedule being changing from day to day, the central clock marker melatonin here is not synchronized. There are exceptions in oil rigs and Arctic stations where people do synchronize, but in most cases, they do not. But shift work is just the tip of the iceberg because we have many other factors that I'll discuss as well. But for shift workers, the most large-scale and long-term data available on chronic disease risk. Now, of course, these data are based on epidemiological assessments, either cross-sectional or longitudinal, so they can't necessarily prove causality to the fullest extent, but they are really indicating that at least some features of shift work are related to this. And other types of situations which are often going together with a degree of circadian misalignment are listed here on the left, which are really just a brief summary. So this stands for circadian rhythm sleep-wake disorders. This is what clinicians would diagnose, but that's only the tip of the iceberg, because those are when patients go to their clinicians, and the clinicians, they say, okay, you meet these long-term criteria, but many shift workers don't meet the criteria for a clinician to diagnose them. And then, of course, there are many other aspects that I don't have time to go into today. I just want to highlight one example, which is jet lag and, social jet lag and obesity, based on a recent publication by Dr. Armand Arab, who's on the call today. And what Armand did is he performed a meta-analysis in about 250,000 participants based on 43 studies to look at the association between social jet lag and BMI, and using many different outcome measures, found that consistently there is a relationship between that. Again, it doesn't mean causality. It may mean that people who are engaged in or have a lot of social jet lag, so the instability of the sleep-wake cycle changing between weekends and weekdays, that may be associated with other factors that, of course, these studies try to account for. So it's suggestive, but not definitive, that this is driving the increased obesogenic phenotype. So if we want to study the effect of circadian misalignment experimentally, we have to think back about what we're trying to do here. And this is one flavor of circadian misalignment, which is the circadian cycle. And here we're really talking about the central clock, for which we have good markers in humans, and the imposed behavioral cycle, so the sleep-wake and eating-fasting cycle. And so to assess whether misalignment of these two result in changes, for example, in impairment of glucose tolerance. And this is from a paper from Chris Morris some years ago. So the first experiment we conducted to investigate the effect of circadian misalignment in humans, and this is misalignment in the example I just said, so misalignment of the behavioral cycle relative to the central clock, what the consequences were there, was from a paper in 2009. What you can see here is the force synchrony protocol raster plot. Some of you may be familiar with this. So in this case, this is very similar to what I just showed you that Nathaniel Kleitman did. And it was actually Nathaniel Kleitman's 28-hour sleep-wake cycle that was refined and now is a gold standard circadian protocol. And why is this such a versatile protocol? Well, because it can dissociate the behavioral cycle from the internal central circadian clock phase. So this is a cartoon of the central circadian contribution to regulation of core body temperature. So this is based on statistical modeling or statistical fitting as for the group average. And you can see that you can now study people where they're aligned. So when they would normally wake up, typically the CBT min, the core body temperature minimum, occurs about 3 hours prior to one's habitual wake time. But you can now study the effect of a misaligned behavioral cycle relative to the central clock in different degrees. And when you look at 12 hours of circadian misalignment indicated here in orange, in response to a similar meal, you can see that the glucose levels rise much higher and stay elevated longer. And also, this is towards the end, despite insulin being elevated more dramatically. But there's also indication here that insulin rise is slower. We studied this in more carefully and in more detail, which is indicated here on right. But again, it's comparing a breakfast eaten at the same time since scheduled wakefulness. But now, instead of eaten, let's say at 8 a.m. in blue, here the people eat it at 8 p.m. in orange. By comparing aligned and a circadian misaligned protocol randomized crossover. So the same person was their own control, and this was much more controlled in this type of experiment. And we basically replicate the finding that we had before. And here we could actually quantify carefully the slow insulin rise, so suggestive of decreased pancreatic beta cell function. And indeed, so this is just one example, but we have many examples where the experimental exposure to circadian misalignment, this behavioral misalignment, resulted in not just impairment of glucose tolerance, and together with Patrick Schouw and his group, we looked at muscle insulin sensitivity being decreased with a euglycemic hyperinsulinic clamp, but also changes in energy balance control, where we also found sex differences. We found increases in 24-hour inflammatory markers and blood pressure, as well as disruption of the endogenous circadian rhythms in oral microbiota that we assess with a concert routine protocol, which is another gold standard circadian protocol, as well as impairments of mood ratings, which link back to the increased risk for depression among shift workers. Now, these studies initially were done in healthy, young, non-shift workers. So then the question came, well, maybe real shift workers are not negatively impacted, because maybe it's a select group or they have somehow adapted. So unfortunately, we found that when we exposed chronic real-life shift workers to this experimental circadian misalignment, we found the same negative effect. The second question was, well, maybe it's just all sleep, because when these people are circadianly misaligned, they're trying to sleep during the day, and their sleep is not going to be as good because of the two-process model of sleep. You may have heard about that. We can discuss that later as well. And indeed, the sleep efficiency is not as good, and the total sleep duration is not as long when misaligned. But when including sleep efficiency and different sleep stages as covariates in the model, the effects we observe are above and beyond any effects of sleep. And this is also supported by work by Evan Kauter and colleagues, where they experimentally tease these two apart. So circadian misalignment negatively impacts these systems, including in vulnerable populations such as chronic shift workers that are exposed to this on a daily basis or a regular basis. So if it's not explained by sleep, might it be that the mistiming of food is actually playing a key role in the adverse effects? And I'll circle back to that later, but remember this question. And indeed, meal timing was a parallel interest of us, which really started based on animal experimental, or was inspired by animal experimental work. For example, this elegant work by Diane Arbel and colleagues in Fred Turek's lab, where they performed a really simple study that remarkably hadn't been done before this time, which is they used a high-fat diet, which is very commonly used in the fields, used as a primary behavioral obesity model in rodents. But what they did is they provided these animals with a high-fat meal just during the active phase, so during darkness in these nocturnal rodents, or during the inactive phase, when they typically wouldn't be eating a lot. And what they found was when they were mistiming the food intake, so providing food only during the inactive phase, these animals gained body mass much more rapidly. And so this raised the question, why would it be, or first, would it be in humans even that timing of food intake, besides what people eat, whether this might play a role as well? And so this is when I met Marta Garolet in 2012. This picture is not from 2012, but I met Marta in 2012, and both of us were very interested in this crossroad between chronobiology and nutrition. And so based on our collaborations and work by other groups, and this is just a very brief summary of references, it was found that late eating is associated with impairment of weight loss success, both with dietary weight loss interventions as well as after bariatric surgery. Then Andrew Maghill, who's now an assistant professor at OHSU in Portland, Oregon, asked the question, is it just the timing of food intake relative to the clock on the wall, so social time you could call this, or is it actually timing relative to people's internal circadian phase, so this phase of the central circadian clock? So what Andrew did is he studied over 100 college students and assessed their dim light melatonin onset, which is a gold standard for assessing circadian phase, and looked at using meal logger, so photographs of all the meals that the students consumed, analyzed those and determined the caloric midpoint in these students, and found that those students who ate half of their calories later, so closer to the onset of the biological night, had a higher BMI and higher adiposity. Now, this is an association study, doesn't show causation, but it's interesting because it suggests that maybe circadian phase is important, which was supported by the fact that when you use clock time instead, so clock on the wall, the association fell apart. So maybe internal circadian time of eating is important. This is difficult to translate to large-scale studies because dim light melatonin onset assessments are laborious and expensive. So Xiang Xiao set out to test whether using a proxy measure of internal circadian phase, which is the timing of one's sleep-wake cycle, may be able to be used to reference time of food intake too. And of course, time of the sleep-wake cycle is something that you can scale up quite easily with sleep diaries, etc. So she studied over 800 participants and found that those individuals who ate later were more likely to be overweight or obese, and she found an interaction with chronotype as well. Now, why would this be? So a calorie is a calorie, so why would it matter when you consume it? So we had some hints from prior work and also ongoing, we're trying to investigate this further. One of which is if you think about the energy balance control, so your weight regulation is primarily driven by the difference between energy intake and energy expenditure. So might it be that if you eat at a different time, your energy expenditure is influenced differentially? And so what we actually had published is that diet-induced thermogenesis is under circadian control. At least we looked at the early stage diet-induced thermogenesis, finding that it's much lower, the amount of extra energy you burn after a meal is much lower in the biological evening than in the biological morning. This was circadian phase determined. And we have found this to be true in a gold standard concert routine protocol that we're working on currently. So if so, then theoretically, if you eat more of your calories late when the amount of energy you burn is less, theoretically, this could contribute to some degree, probably not a large degree, but to some degree to increased risk for body weight gain or less body weight loss if you're dieting. The flip side of that scale would then be the drive for food intake. And also there we had previously published work showing that hunger and appetite are under circadian control with a peak in circadian hunger drive in the evening hours around 8 p.m. So maybe also, depending on when you eat, maybe you would overeat more likely if you're eating later. So with that question, we set out to address this question, what mechanism or mechanisms may help explain the increased body mass when eating late in the day. And so the three mechanisms we explored was whether this might lead to an increased drive for energy intake, so increased hunger and appetite, decrease in energy expenditure, or changes in molecular pathways in the main storage organ, which is fat or adipose tissue. So this study was conducted by Nina Vujovic, who had people come back to the laboratory twice. So this was a randomized crossover study. Again, people were their own control. Here you can see an image of the laboratory. Before each in-lab protocol, they maintained a fixed sleep-wake cycle for about 3 weeks. And in the last 3 days prior to their admission to the laboratory, they got all the food provided, and they were required to eat this in a particular time window. So the early eating protocol that's displayed here in the middle, you can see the meals, how they are timed, and the meals are interspersed by 4 hours and 10 minutes apart. In the late eating protocol, they received, again, identical meals on these test days. Test days are indicated on day 3 and day 6. And on the test days, we also assessed their hunger and appetite scores, their leptin and ghrelin concentrations in their circulation, their energy expenditure with indirect calorimetry, their core body temperature continuously with rectal temperature sensors. And also on day 5, we collected a abdominal subcutaneous adipose biopsy after an identical fast duration in both protocols, so we could compare the molecular pathways. And at the same time, we controlled for everything else, so for the caloric intake, dietary content, fasting duration, etc. This was a strength of the protocol. The obvious limitation of this protocol, it's short term. So we only assessed this for a duration of, well, acutely, and then after 4 days, we repeated to see if there were any changes. What did we find? So for the drive for energy intake, when looking at subjective ratings, this was using visual analog scale. So these are computerized tests where they click on a line to represent their degree of hunger. You can see in red is the late eating condition, and in black is the early eating condition. And the vertical lines are indicating the meals. The black vertical lines are when the meals are given for the early, and the red vertical lines for the late eating condition. And so what we did is we analyzed this. We found no differences, no statistically significant differences between test day 1 and day 2, so day 3 and 6 in the protocol. And so we combined these to look at the likelihood of people reporting hunger, which we defined as higher than the 50% mark in these people. So we found a significant increase in hunger in these individuals. And this matched the objective measures then. So hunger was subjective. We also looked at objective measures, leptin and ghrelin. And for example, here we looked at the ghrelin to leptin ratio and found that both the wake time and the 24-hour ghrelin to leptin ratio was elevated. And this was active ghrelin, so acylated ghrelin. So consistent findings both from subjective and objective measures for hunger intake drive. What about energy expenditure? So we used indirect calorimetry with ventilated hood during the wake episodes every 1 or 2 hours. And what you can see is that here, energy expenditure is actually lower during the wake episode. Now, we couldn't assess energy expenditure during the sleep episode because this is by a ventilated hood and people can't sleep under that. But we used core body temperature as a proxy measure, given the fact that the environmental temperature was constant and their layers of clothing was the same and their physical activity was the same between the early and the late protocol. And what we can see indeed during the wake episode, you can see similar reflection of this decrease in energy expenditure with lower core body temperature. And this was not compensated with higher temperature during the night, if anything, even during the sleep episode, where they were immobile, etc. Temperature was still lower. So again, consistent measures, consistent findings with both these measures indicating a decrease in energy expenditure. Now for the adipose biopsies, so this was really Matthew Brady his expertise. And so what Matthew did is he analyzed the biopsies. We sent them to Chicago. He's at University of Chicago, and performed pathway analysis, finding increase in adipogenesis pathways and decrease in lipolysis pathways. So again, consistent findings on different pathways, suggesting an increased pathways for fat growth. So while we set out to test which of the three pathways might be explaining this increased risk for obesity with late eating, we, to our surprise, found that all three pathways are actually involved and pointing in the same direction. Just to emphasize again, this was a short-term study, but it was meant to test mechanisms involved. So the other interest we had was regarding glucose control, as I briefly discussed before. And in 2009, I was lucky enough to meet Richa Saxena, who at that time had just published a number of papers to show the discovery of a new risk factor for type 2 diabetes in the melatonin, one of the two melatonin receptor genes. So MTNR1B, which is a gene for the MEL1B or MT2 melatonin receptor. So this risk variant Richa had reported on. And so at that time, we had just published our circadian misalignment work. So we were very interested to collaborate and see to what degree melatonin signaling may be relevant here. Now, the GWAS, the genome-wide association studies, you should realize that those studies that this discovery was based on was assessing glycemic control during the daytime hours, because that's when the clinicians are working. And so for most of those glycemic measures, you're assessing glycemic control when melatonin is absent. So it's like studying a receptor without its ligand present. So our question was, if you introduce the ligand, so melatonin, for the receptor, might you find a more clear-cut effect? And could we leverage this to study the role of melatonin and the MT2 or MEL1B receptor in glucose control? The other thing just to mention is that this got quite some attention, and the diabetologists started to really pay attention to circadian biology. So I think that's a big win for our field. And in part it was because this SNP is really common. The single nucleotide polymorphism occurs in about 50% or so of the people attending this lecture. And so what we found that melatonin impairs glucose tolerance when we introduce it, and that it's exaggerated in the carriers of this risk variant. And we looked at this initially when we provided people with oral intake. So we gave them 5 milligrams of melatonin, which you can buy over the counter without prescription in the US. We also wanted to know whether physiological nighttime concentrations might be relevant in this signaling. And so the way we approached this was to look at early versus late dinner timing. So dinner either when melatonin is still low or when it's already elevated, and found that this was worse in the glucose tolerance when eating late, and it interacted with the MTNR1B risk SNP. Now, that was repeated, that last study I showed, in a much larger scale. And this was really driven by Marta, who enrolled over 800 participants and randomized them to either an early or a late evening OGTT, so to simulate early or late dinner timing, and found that the late eating in black here compared to the early eating in gray here, and again, this is within subject comparison, resulted in a worsening of glucose tolerance, or so with other words, glucose levels rose higher and stayed elevated more. But this effect was stronger in people who are carriers of the risk SNP, and even more strong in people who are homozygous for this risk SNP. And you can see here this dose response relationship with the number of risk alleles that these people had. This also went hand in hand with a decrease in the insulin released following the OGTT. So indicating a decrease in glucose tolerance and suggesting at least an involvement of decreased pancreatic beta cell function, especially if you are a carrier of this risk SNP. So just to say that based on this work together, we would suggest some caution with eating either close to oral intake of melatonin or close to the biological night when melatonin is elevated. So now the last part, can we actually now utilize this knowledge about meal timing and the adverse effects of circadian misalignment to test whether, at least in principle, meal timing may be advantageous as a countermeasure against the adverse effects of shift work? So this is basically this question. So can we prevent or at least minimize the adverse effects with meal timing? And again, we were inspired by animal experimental work. This is work by Damiola and colleagues, in which they in mice took out the liver and looked at this at different I think these are CT times, so circadian times, so on DD. Although we will see if I'm right here, maybe it's CT. Regardless, so as you have heard from previous lectures, so CT time 12 is when the animals typically become physically active, and that's CT 0, they start their rest phase. And you can see that in this case, the liver per 2 rhythm peaks at 22 hours when the animals are fed during the nighttime. But when they're fed during the daytime, their liver clock, the molecular clock phase, shifts by about 12 hours. So they completely shift the timing of their clock in the liver. What happens to the SCN? What happens to the central clock? Nothing. So the central clock, either during nighttime feeding or daytime feeding, shows the same rhythm in per 2 as a readout of the molecular clock. And so what this therefore shows is that mistiming of food can result in internal circadian misalignment. So misalignment between different molecular clocks throughout the body. So that was one inspiration. The other inspiration was work done by Salgado Delgado in the lab of Carolina Escobar, who kindly provided this video you see in the top left, which is the design of a rodent shift work model. So what they did is they had animals in these slow rotating drums. You can see they're not running, they're just gently walking. What it really means is that when the drum is rotating slowly, the animals cannot sleep. So this is the simulation of having to stay awake at your job. And so what they found was that if the animals are provided with food ad lib, then in the work conditions, so the W here is where they are required to work now during their habitual rest phase, so they're engaged in shift work, if you like, or night work for them. I mean, for us, it would be night work. For them, it's day work. And what you can see is that the body weight as well as the adiposity increases. And this was shown by others as well. What was really nice in this study is that while these animals were eating while these animals were exposed to the simulated night work, the animals were now experimentally restricted of food, and only the food was given during their normal active phase. And when they did this, so this is feeding during the night, so during their active phase, you can see that even though these animals are still engaged in shift work, when the eating rhythm is normalized, these animals revert, or at least partially revert, their metabolic phenotype. So with other words, this experiment suggests that food timing is actually really important in this simulated night work protocol.
We have 5 minutes left.
Thank you. So the questions therefore were, first, can behavioral misalignments, so as a model of night work, cause internal misalignment also in humans? Second, can circadian daytime eating during this circadian misalignment, so the misalignment of the sleep-wake cycle, if you correct the eating time, can you first reduce this internal circadian misalignment? So can you help get these internal clocks realigned? And second, can you improve glucose tolerance that we had shown to be negatively impacted with circadian misalignment per se? So this is the protocol that Sarah Shalapa conducted. Sarah conducted this when she was a postdoctoral fellow. She's now an associate professor. So in this protocol, this was a randomized parallel group design. So people were either randomized to the control protocol on top or the intervention protocol on bottom. The one part of the study you will recognize, this is the force of synchrony portion between days 7 and 11. But what we also did is on days 3 and 12, we started a constant routine protocol where people were kept awake continuously for this whole duration. They were remaining in bed at rest. They were in dim light at about 3 or 4 lux, and they got isocaloric snacks every hour. And so by doing this protocol, the constant routine protocol, we can extract the endogenous circadian control of physiology. So we wanted to assess this at baseline, look at the effects of circadian misalignment with the force of synchrony protocol, and look at the after effects if the circadian organization was modified by prior history of circadian misalignment. And the only difference with the intervention protocol was that the eating fasting schedule was locked in at a 24-hour cycle. So we disconnected it from the sleep-wake cycle and we aligned it with the central circadian clock that ticks along at a rhythm of very close to 24-hour cycle. So the hypothesis was that we could help minimize adverse effects by normalizing or synchronizing the eating schedule closer to the central circadian clock. This is what we found. So on top, we can see that the central clock doesn't really care. So the black is the first CR in the protocol, the orange is the second CR. So after misalignment, on the left, the control group, on the right, the intervention group, you can see that the temperature rhythm is not significantly different between these two, between before and after the misalignment in neither of the two protocols. But what happened to the glucose endogenous circadian rhythm assessed during the constant routine protocol is from before in black to after the force of synchrony. The endogenous circadian control of glucose was completely inverted. And you can see that in the intervention protocol, this inversion was prevented. And this is indicated here in this radial plot as well. Interestingly, if you look at insulin, in both protocols, insulin rhythm became non-significant. If we look at the effect of circadian misalignment during the force of synchrony, so while eating, let's say at 8 a.m., here a breakfast, and at 8 p.m. at dinner in black, versus an inverted sleep-wake cycle here in orange, you can see that glucose tolerance is impaired and insulin levels are low during the first portion, indicating an impairment of pancreatic beta cell function. In the control protocol, we could fully prevent this in the intervention protocol just by normalizing the eating fasting schedule to 24 hours. Interestingly, we also found that the glycemic adverse effects were stronger in those people who had stronger internal circadian misalignment. Now, this is just an association. It's not causality. We can't conclude that the internal misalignment causes the impairment of glucose tolerance, but it certainly is an interesting hypothesis to test on future studies. So to summarize this last part is that yes, we can prevent we can induce internal misalignment in humans. We can prevent this by the meal timing intervention, and we can also improve and prevent these adverse effects on glucose tolerance. And just to say that we have a number of other outcome measures from this protocol, one of which is already published just 2 years ago on mood. So take-home messages, first is that circadian misalignment causes adverse cardiovascular, metabolic, immune, microbiota, and mood effects. Second, that food timing affects energy balance control and glucose control. And so energy balance control involving 3 mechanisms, both on energy intake, energy expenditure, and adipose molecular pathways. And for glucose control, I've only shown you the melatonin-related work, but we have other work as well. But for the melatonin-related work, we find that eating when melatonin levels are high impairs glucose tolerance, especially in MTNL1B risk carriers. And then finally, food timing is at this stage a promising countermeasure. More work is obviously needed, but a promising countermeasure against adverse health effects. Not just on glucose control, but other factors as well, and possibly, but that requires further study through restoring internal circadian alignment. So with that, this needs no introduction to this audience, but in the behavioral field, nutrition and exercise have been receiving all the attention. So one should also add sleep to this. But of course, all of these behaviors, you should also think about this in a temporal framework, because it's not just what you do, but also when you do it. So with that, I would like to thank all my collaborators, colleagues, funding agency, and you for your attention. Thank you.
Thank you very much, Frank, for the great talk. I will definitely not eat late anymore.